So hi, Luca. We'll get going. So you have 2 decades of experience in the gene therapy field with recent focus on in vivo engineering of human hematopoietic and stem progenitor cells. How have you seen the gene therapy field evolve since your first involvement, and what have been the key milestones along the way?
Well, first of all, thank you, Nick, for having me, and it's a pleasure to talk to you and Anson Wade about my experience here at Envelop and my previous experience in gene therapy. Yes, you're right. I've been in the field for quite a while now. I started as a PhD student in Milan, and at that time, I was lucky to really touch firsthand the pioneering work of many great people in the field of gene therapy in Europe. In particular, I'm talking about the work that Luigi Nardini and Alessandro Iuti developed in Milan, particularly, and also to follow also the interaction I was able to have with other pioneers like Christopher Baum, Adrian Trasher, Bobby Gaspar, and then later expanding my network in the US with David Williams, Don Kohn, and many others. My initial approach for gene therapy, my initial work was based on studying the safety of lentiviral retroviral gene therapies. And particularly, I worked a lot in ex vivo gene therapy field for primary immunodeficiencies. And this was an area that was entering the clinical stage when I joined the gene therapy field in 2006. And we were getting very early results that were showing efficacy and safety of this approach to cure kids with these inherited disorders. And this was the first proof of concept that gene therapy worked and could be a curative treatment. In parallel, the AV field was developing in vivo, the early in vivo approaches with very good results in ocular diseases and hemophilia. And so essentially gene therapy was opening up to the world, and we were in a phase where the technologies were moving from the bench to the bedside, and we got the first exciting clinical results. It was great to experience that phase, that pioneering phase. I remember still the meeting were pretty small, and where there were a lot of big names sitting at the table with me, just a simple PhD student. It was great to be part of that growth in the field. And later on, eventually, the main milestone that came in was the availability of a editing platform, and particularly the advent of CRISPR-Cas9. And from that point on, really, the gene therapy field boomed. And there was more and more interest, particularly from the private sector, in participate actively, not only in the commercialization of the products, but also in the research. And so I've witnessed that over the past decade. And now we are at a point where we're trying to combine the early experience in the early days of gene therapy in patients and with the new technologies and try to really make gene therapy accessible to the broadest population of patients possible. And this is exciting because now we're kind of moving to a phase to make a parallelism with the world of computing, where you went from mega computers in the 50s to the personal computers at home. And now we are in the phase where we're switching to the mobile device. And so if I can use that metaphor for gene therapy, that's the exciting phase we are witnessing right now, is like the ability of really producing these therapies cost effectively and making them accessible for as many patients as possible. That's what I'm glad to be part of.
And part of that revolution currently sits within the gene editing field, and specifically lots of focus at the moment and discussions around delivery, and thinking towards in vivo delivery. Lots of people talking about this as the future of the field. What are, in your opinion, the current limitations to widespread adoption of in vivo delivery for the gene editing field?
Well, this is indeed an important aspect. Actually, was already at the top of the list of the important things to address from early on. Remember the early days in the AAV field where pioneers in the AAV world were mentioning the fact that it's all about delivery. And we are back to that because we did a lot of progresses in terms of the platform, as I said, the editing platform, but now we really need to put these tools into a delivery vehicle and get into the patients effectively. So one of the key aspects that come to mind in terms of what we need to solve in that space are the fact that we definitely need programmable tissue specificity. So we need to be able to generate particles that not only biodistribute well in the patients, but avoid organs that you don't want to target or tissues that you don't want to target and really deliver the gene editing cargos to the cells that you want to correct. So that is a critical aspect, and there are different platforms and there are different companies that are working on that, including Envelop. The other key aspect is the cargo diversity. Ideally, you want to build a particle that can accommodate different type of cargos, different type of editors, potentially targeting multiple sites at the same time, and building that particle in a way that is as modular as possible is critical for the field. Scalable manufacturing is the third aspect, right? You want to make it scalable and you want to keep it cost effective. And eventually this translates to cheaper therapies for patients. So it's very important that we get to that point where we can produce these particles at scale, and that we have this in mind from early on when we're developing this platform. Of course, safety is important. I worked on safety a lot in the ex vivo gene therapy field. In that space, we were infusing a combination of a cell and gene therapy product because essentially we were infusing back engineered cells. Now we're talking about delivering particles directly into the patient. So it's important that we prove that we avoid the targeting for example, reproductive organs or other areas that might have a detrimental effect for the patient. Of course, what I think Velo brings to the table is particularly the advantages related to the ability to retarget our particles to certain tissues by playing with the outside of the particle, and also deliver multiple cargos for multiple indications. So what we want to do is really build that modular particle that we can use for multiple targets in vivo. And eventually, that's where the whole field is heading.
To bring some of this to the present day, I'm aware that lots of folks listening to this will be very well aware of your team's work, but just for background, Envelop Therapeutics are developing and applying to efficient and cell-specific in vivo platforms to enable, like you mentioned, the delivery of a range of cargo to target cells. Could you tell us a bit more about your own role leading the particle development team and where your team's current focus areas lie?
Sure. So I'm leading the particle science team here at Envelop, and we are developing, we're working on two main platforms, as you mentioned. One is we call it the DeliverX. It's essentially a viral-like particle that is built with a bilayer lipid envelope on the outside, and then a capsid and a cargo on the inside. And the cargo can be a complex ribonucleic protein, and we can accommodate Cas9 editors, prime editors, and different type of other editors. But we are also working on accommodating other type of cargos. And the second platform is a sort of modified version of the DeliverX, where essentially we are working on a particle that is a simplified version of it, is called DeliverM, where we are trying to fuse our cargo directly to the inner leaflet of the membrane through a plextin homology domain. And this way we don't need the capsid, and we remove one viral component from these viral-like particles, therefore reducing potential for immunogenicity. So we want to try with this version of the particle to create a fully humanized delivery system that has human components on the outside and on the inside, so that it's better tolerated for in vivo delivery. So we're working on both systems. And what my team is doing is taking these systems and trying to work on the different components of each of these particles. From the outside, we're trying to engineer the envelope so we can make it specific to certain target cells. We are also trying to work on the inside, so making a better capsid and also accommodate as much cargo as possible to achieve the best potency upon delivery. And that's basically my teamwork, is trying to work on optimizing these particles. And then I'm also leading a program that is aimed at targeting this particle to hematopoietic stem cells in particular, because that's why because I'm building on my previous experience on hematopoietic stem cells in the ex vivo gene therapy field, and we are getting very interesting early results that we hope we can share soon, including at Hanson Wade, the upcoming Hanson Wade meeting.
Teed me up perfectly for the next question. So at the fifth Genome Editing Therapeutics Summit this year, you'll be participating in a panel discussion that focuses on delivery vehicle selection for extrahepatic disease indication targeting. And as a sneak preview for some of the more forward-looking topics, sorry, to be discussed as part of this panel, where do you see the future of gene editing delivery going?
Yes, absolutely. No, I think pretty much touched on it in terms of the future is the ability to really deliver efficiently gene editing cargo in vivo where we need them in the human body. This is the holy grail of gene therapy, essentially. And this was already from the pioneering phase of gene therapy what we really wanted to achieve. But we didn't have the technology then, and now we have it, and we need to make it work. So the future is definitely in vivo delivery systems for gene therapy. I'm glad that I'm part of a company that's working on building that future. And we're coming from different angles, right? Different companies are using different particles, and that's the beauty of it. That and also as it worked well in the early phases of gene therapy, where we had different viral vectors and technologies that we were testing, for example, in ex vivo gene therapy, and they all came to fruition at more or less at the same time, and we had the chance to test them in the patients. I foresee that this is going to happen in the in vivo gene therapy field, and we're going to see a boom of more in vivo delivery platform reaching the clinical stage and eventually being tested. And we're going to learn a lot from that. What I'm saying to the rest of the field, both on the academic and on the industry side, is don't be shy to be the first going in. I understand sometimes people are concerned about burning their technology, but really now is also a matter of asking the investors to have the serendipity to invest on these platforms in the long term, because that's essentially the end game for gene therapy, is being able to develop this platform. So I don't see any other innovation that is more important than this at this point. So we need to really give the chance for both small biotechs, big pharmas, but also the academic field to really develop these processes and test them in pioneering work in the clinic, as we did in the early phase of gene therapy.
And finally, why are you excited to be involved in the summit this year?
Well, I think first of all, thank you again for the invitation to the summit. I think for me, the great thing about it is, as I said, the fact that different industry actors in particular are coming from different angles on the same program, on the same main goal to deliver, to achieve efficient delivery in vivo. And I'm curious to see what they're up to, essentially, and sharing my experience there, and in a collaborative fashion. More and more people, I realize that people really want to make things happen in this field, and that we come in with an open mind, and we really is much less about competition in this meeting, but more about collaboration and networking and understanding how we can really leverage each other to for the best of the patients. Eventually, what we want to do is make an impact in treating diseases and allowing this therapy to reach the broadest patient population and making these therapies accessible and cheap enough so that they really become an alternative reality in the clinical world. So I think it's all about that eventually. So I'm excited to participate to this meeting and to share and share our experience, develop and collaborate with other colleagues in the field to really make this a reality for patients.